Why this is so cool is because all those little dots are elements of this brain. Neurons. There's a lot in here, and this represents decades and decades worth of work. And I wanted to go into this because this is a really, really cool study. But there is a fruit fly brain, and it's what is called the connectome that we are looking at. So this is basically a map of every single neuron in the fruit fly brain and how it touches another neuron. So this is 140,000 neurons in this brain. 140,000 neurons are mapped out. Fishman, those colored dots, they're pseudo-colored. So this is just adding in the extra visual that you can actually see the bodies of the neurons. But in this 140,000 cell mass here, there are 50 million connections. So that means that every single neuron isn't just connected to a single other neuron, right? Because that would be what, 140,000 times 2, maybe times it 2 again, just for the sake of the 2 connections. No, it's 50 million connections. They've already done these kind of images before, and we talked about it last year on Science News. The most other complete one with that was a fruit fly larval brain. So the fruit fly larval brain has about 3,000 neurons. And so this is a big step up. But basically they took similar tech that they did in the fruit fly larval brain and just, upped all of it. To give you some perspective as to this brain is less than 1 millimeter wide. I know it's blown up massive behind me, but when you're looking at it under the microscope, it's less than 1 millimeter. They took one single female brain, and this is where lasers come in. They took that single brain and cut that brain into 7,000 slices. The cutting was a use of combination of a diamond blade and lasers to make, because imagine, again, you're making something 1 millimeter wide into 7,000 slices. And each one of those slices is 40 nanometers in thickness. So it's thinner than your hair to get these slides to be able to do. And then what you ended up doing was you put them into that scanning electron microscope and took an image, all 7,000, right? There's no way that you can stitch together these images of a single person and make this map come out. So what they ended up doing was they built AI. They had an AI system to start stitching stuff together. The problem is that this AI is not great because you're looking at such a small scale. So Fishman, it still made a bunch of errors. So the AI was a starting point, but then they had 287 scientists and general public volunteers manually linking the images together. So imagine, I'll show you some videos of this. This image is taken when you're going through layers upon layers, right? 7,000 layers you're going through in this image. So imagine that one person takes those 7,000 images and just tracks one neuron. In this image, okay, you circled, here is the neuron I'm interested in. Next image, this is still that same neuron. And then you keep going like that. And so when they estimated how much time that this took between all the people, they said it was 33 person lifespan years that this took, which is, it's kind of wild and on there, right? So what's the next step? And how are they using it? So that's a great question, Jay. So remember I mentioned that this was only done in females. This was only done in females. And that is a world of difficulty because only being able to do this in females, we've talked about many times here, right? That if you only use one of the sexes, you're going to be missing half the picture, no matter what. And so their next plan right off the bat is to do this in males as well, so you can have the sex differences and compare those. Now, after that, they need to figure out what neurons talk to another neuron and, make the behaviors for it. So embedded in these images, you've annotated all the neurons, which means imagine that I drop you in New York City and you're the only person there. I ask you to draw me a map of the entire city. And so you go and you draw a map with streets using the street signs and buildings and building names. That's basically what they've done in this brain. Okay, but that doesn't tell me where in New York do I go for a great pizza, or where in New York do I go for a great show or a baseball game, right? Because all you've given me is the actual streets and signals. Maybe I know the stoplights as well, but I don't know how to get there. So now you need to figure out how to trigger something like that in a brain. So instead of just having the Google map you now need a Google map that can actually take you to a location. Does that make sense? So again, this is just called the connectome. This is like all the connections from it. And now what you need to do is try to simulate how the fly brain works. And so that's actually using some of the experiments that we've talked about before, where you shine lasers on the fly's brain and trigger it to do particular behaviors. Now that is much easier to trigger, because let's say like I show this fly something that it's scared of, like a parasitic wasp, right? Like, and you see some of the neurons have changed color using some of the tools that we have. And when you end up having that, it's like, okay, well, I can maybe ID that there's some activity here in the brain, right? But maybe, Jay, now that I have the connectome, I can be like, okay, I know up to this point, but I also know that at this point, this cell connects to this one and this one and this one and this one. So you can almost like extrapolate and trace inwards what exactly is going on in the next level. And so it gives you a roadmap. Like basically, Jay, if I take you to the edge of New York and I tell you, okay, here's step one of your journey to get to that pizza place, and I direct you for step one, but at step two, you're like, where do I go now? Well, I've given you the map. And so then you can be like, okay, well, I know I'm here and there's only 2 other streets that I can go down. So I'm going to try one of these streets and figure it out. And so it's almost like you now have a little bit of a puzzle map that you can use and figure out how this works. Does my pizza analogy make any sense, y'all? Hopefully that makes some level of sense. Again, I think it's really, really cool, Jay. Now, right now, I would still say that this is much more of I have the map of New York, but I don't know yet where your pizza place is. And then after your pizza place, you want to get back home, like wherever the output is in the body, right? That next neuronal connection. And that I don't even have a map for you. I don't have anything for you on that. But it's definitely that first step in where this is heading to. Yeah. I really, really like this figure. So this, y'all, breaks down each region of the fly brain. And so it's in different colors here, but what you can see is the retina is in one color, and then the optic lobes are broken down into different colors as you're getting closer in. There is a fan-shaped body here. There's the ellipsoid body. There's the mushroom body, which is learning and memory center. There is the antennal lobe here in blue, which is connected to the antenna. There is a subesophageal ganglion at the bottom. So there's all these independent structures in the fly brain. And one sanity check in their data was that each of these sub-regions are different in terms of how they're actually comprised. What neurons are there? What types of neurons are there? You would expect that based on just a prediction, but to actually get that as an output is really, really cool. So it's like a confirmation of what's happening, like in terms of it. Fruit Loops, well, corn, we were actually looking at the video that was mapping out like individual units of it. So here is just like the visual center. But yeah, here is just looking at resetting back to the brain and the neurons and all the connections to it. So they took that whole image of the entire system and they broke it down by different centers and compartments as well. So if you just wanted to look at visual system, there are the visual system neurons alone. You can see all the connections, the rhabdomeres going in, all the different cell types connecting within. This is where all the projection neurons from the visual system come in, so the visual processing, visual centrifuge elements to it. Here is the central body of the fly brain. Ascending neurons. Oh, because it's cool. Descending neurons. That's the feeding zone, so mouth movement for motor. Endocrine system, so these are the endocrine producing neurons. So you can even figure out what neurotransmitters are made by these neurons too. So like, while this video is what, a minute long, it 6 years of work. So there we go, chat. That is, I think, our nice summary of the fly connectome. Second paper was the entire connectome of the fruit fly brain and everything that encompassed with that, like what that means, which I think, again, was a lot of fun to talk about. It was a super cool paper, and I hope you all enjoyed it as well.